Gentlemen, we are moving forward to our next session, which is on disruptions and digitization trends in clinical research. I would like to invite Mr. Manoj Saxena, Managing Director, Bayer Zydus Pharma and Country Division Head, South Asia Pharmaceuticals, to come and introduce and welcome our keynote speaker. A very warm welcome to you.
Thank you, Kathy. I know we are slightly delayed, so let me quickly, do a warm welcome to Christoph, who is a dear colleague of ours at Bayer, and also introduce to everyone who Christoph is. So Christoph is currently heading Bayer's global development and clinical operations, overseeing clinical development, including executing clinical studies globally. Prior to Bayer, Christoph had leadership roles with Otsuka, Bristol Myers Squibb, Novo Nordisk, and a few other companies in the field of medical and development functions across globe. Christoph received his medical degree from Rupert Karl's University, Heidelberg. He also holds an MBA from Schiller International University and is board certified in internal medicine and diabetology. And as you can see, being a doctor coming from medical background, he's primed his ability to combine scientific R&D innovation with a very distinct strategic approach to business development opportunities, regulatory navigation, and global patient access solution. Extremely delighted to host you, Christoph, and a big thank you on behalf of OPPI and all of industry colleagues in India. Looking forward to your keynote address. The spotlight is now on you. Thanks a lot.
Thank you, Manoj, and thank you for this very kind introduction. I'm truly delighted to be here and share a little bit what we are doing in Bayer in the end to change the way we run clinical trials to get our innovation to patients earlier. If we go to the second slide of my presentation, please, since we had the introduction already. One slide more, please. What I want to focus on in my discussion with you today, if you proceed to the next slide, please, is what technology are we using in Bayer? What are we doing in Bayer to achieve what we want to achieve, which is make sure our innovation reach patients as fast as possible. The way we conduct clinical trials, on the one hand, has to ensure that we achieve the highest level of quality when it comes to our study data, but on the other hand, get meaningful results as fast as possible. And we do think that technology, specifically digital technology, is a way to go that can actually help us achieve that. And what we're doing here at Bayer is that we have focused on certain activities around the clinical development process that helps us to achieve that. And I will now go to each of those activities a little bit more in detail, and some of them actually I will hone in on. The first thing, when we talk about using technology, using digital technology specifically, but technologies in general, actually it's applicable as well, is that we have to make sure that we create a landscape that enables us to use different technology together. And the reason why we call this a study management landscape is because it is a landscape and not just one system, because it's different systems, it's different technology that we have to combine and make to work together in order to utilize the newest and best technology that we have at our disposal. We are focused very much on making the right decisions. Quantitative decision-making is an important part of helping us to make these decisions. We have a lot of data, we have a lot of information at our disposal, and we have to make sure that we, on the one hand, analyze that data correctly, and on the other hand, making sure that we take the right decisions out of this and use advanced analytics and data insight to obtain these decisions. In order to use advanced analytics and data insights, we have to make sure that we have the right platforms in place that can collect and store the vast quantity of data that we have and the even vaster quantity of data that we will have in the future, and then have the right platforms to ingest and analyze this. A lot of activities around the clinical trial process have traditionally been very manual. There's obviously the problem with using humans in some of these activities in respect to time. It takes more time, but there's an important quality aspect here as well, that if you introduce human error, then obviously you jeopardize quality. By automation, we are trying to remove this error and increase this quality and basically deliver what we want to do at a higher quality as well as in a shorter period of time. The quality and the kind of data that we are collecting is changing. Real world evidence has become a little bit of a buzzword, but being able to collect data that is much more real world like than what we traditionally have done in clinical trials is extremely important to collect data that is meaningful for healthcare professionals, for patients, payers, and governments. And we have to make sure that we have the ability to appropriately collect that data, to appropriately analyze that data, to appropriately interpret that data so that it can help us making important decisions. We have increasingly used patient registries, not just to collect outcome data, but to receive information around where are patients located, what centers are treating patients, in order to make sure that we place our clinical development studies in the right location in order to obtain a patient population in our clinical trials that is representative of the overall population that will be treated with this asset in the real world. And we have been using what we call decentralized clinical trials increasingly in order to make it easier for patients to participate in clinical trials and reduce the burden that clinical trials provide to patients. And let me now go to the next slide and talk a little bit more about what we are doing with decentralized clinical trials, or DCTs. The idea of a DCT, or decentralized clinical trial, is rather to bring the patient to a center, to actually bring the study to the patient. So what we're trying to do in DCT is to locate as much as possible any study-related activity around the patient. That means that rather than the patient going somewhere to be examined, we're trying to doing examinations at home. This can happen through home visits of a physical person, but this can happen with digital devices, like, for example, a blood pressure cuff that then sends a measure to a database. This can happen through, for example, lab draws at the home of the patient, or it can happen by the patient collecting a dry blood spot and then mailing that dry blood spot off to a lab. But whatever is feasible and possible, you try to conduct at the patient's home as much as you can. What you achieve by doing this is that obviously the overall burden of the patient in participating in a clinical trial, having to go somewhere in regular intervals, having to wait, having to come back, you reduce that. You eliminate a lot of anxiety that comes around, and specifically in the last 2 years has been a huge problem of not wanting to go into a healthcare facility, and limit the interaction that the patient has to have with a healthcare facility, with humans in healthcare facility that might stress some patients out specifically in a pandemic situation. You can use home care facilities that already exist, like home care nurses, for example, or you can use completely new devices, new technology, like, for example, a medication dispenser combined with a blood pressure cuff, an ECG monitor, that where you can upload data to a platform that basically enables you to collect information. You can tailor this to any study, any endpoint you want to collect. You can, the use of technology is infinite and only limited to whatever technology is available at the time. So the whole idea of decentralized clinical trial as a concept is not going to change. But what is going to change is that the technology you can use to successfully collect data in DCT is constantly going to evolve, and therefore, of course, going to change the face of DCT over the next coming years. I want to say one more thing about DCT before I move on, and that is obviously the way we conduct clinical trials for pivotal studies, for regulatory grade clinical trials, is of course driven by the quality demand that regulatory authorities have on clinical trials. The acceptance right now of DCT collected data for pivotal studies varies dependent on which regulator you talk to. And there is obviously skepticism about making sure that the data you collect, the endpoints you collect using DCT, has the same degree of quality than our traditional brick and mortar setup. And that is the reason why you do not see right now completely DCT run pivotal studies. However, I would like to add here yet, because as our experience with DCT grows and evolves, as our confidence in DCT grows, I personally think that the acceptance of DCT studies for pivotal studies will grow. However, it might not be a black or white scenario where you will have a study that is done 100% brick and mortar and 100% DCT, but we might find ourselves in a situation where we have a certain percentage of patients that will be participating in a study via a DCT approach, and then you might have a certain percentage of patients that will participate in a study via brick and mortar site. The advantage of having this flexible approach, of course, is that you can make the participation in a DCT approach dependent on the specific patient's living situation. Yeah, how is the access to technology? How is the access, for example, to data connection, and or how is the access to home care? Yeah, and you can take a very flexible approach. If you go to the next slide, please. I already talked a little bit about using metadata-driven optimization to increase the efficacy in how we run our trial process. I talked about the fact that obviously there's a lot of manual work and labor involved in this process right now that takes a lot of time, and that on the other hand introduces quality risks. If we standardize and automate this as much as we can and automate the generation of different documents, being it part of the protocol, being it part of the study report, as much as we can, then we achieve two things. The quality of the documents that we produce will improve, and the time it takes to produce these documents will be much less. And that is the reason why we are constantly trying to automate this process as much as we can. And as technology evolves, and here specifically artificial intelligence plays a very important role, our ability to do that increases. And that is something that is probably not as visible as, for example, conducting a decentralized clinical trial, but it is an equally important improvement in order to obtain what we want to obtain, which basically is improve the quality and the speed of our clinical development projects. And that's why this is so important. If you go to the next slide, please. The other thing I already talked about is that the volume of data that we are collecting already is extremely large. With the use of digital technology in clinical trials alone, the volume of data we are collecting in clinical trials is going to explode. Just imagine I was talking about at-home monitoring of, for example, 3-lead ECG. What if you have a patient wear this for a long, extended period of time? What if you start monitoring sleep, breathing pattern, pulse pressure in your clinical times on it over a certain period of time? Imagine the amount of data that you have to collect and store and process in order to do that. And that is the reason why it's extremely important for us to create these advanced analytics and data ingestion platforms in order to handle that. So it's our ability to store and collect the data. And then, of course, our ability to process the data, and then making sure that we analyze the data in an appropriate way to then draw the right conclusions. And with the increasing amount of data on the one hand, and the importance of making sure that we can rely on the output here, that has been a huge focus of our organization as well, to ensure that we keep up with the demand that our advanced way of running clinical trials puts on the systems that we have. If you go to the next slide, I want to give you now some examples of how we're utilizing this in a specific development program. Before I do that, I do want to highlight that we in Bayer have just been ranked the second place out of 53 companies in a site survey looking at the site relationships conducted by CenterWatch. The reason why I want to share that with you is not only because I'm proud of it, because yes, I'm very proud of that. But the other reason is that I personally believe that one of the reasons why we have such excellent site relationships is the way we try to ease the process of running clinical trials as much as we can. And I think technology is one big advantage of that. The program that I want to briefly share with you, where we utilize our innovative clinical trial approach, is a program where we look at a new anticoagulant in patients, for example, with atrial fibrillation. If you go to the next slide, go back. This program is a program. If you would go back, please. Sorry. So this program is a program where we look at patients that have atrial fibrillation, and where we use some of these approaches in the conduct of the study. So what we are doing and have done in this program, for example, is that we have a certain percentage of patients where we use DCT. We are currently conducting that in the United States and have an agreement with global regulatory authorities that we can use a certain percentage of patients in this program where we collect the data using the DCT approach. The other thing we have done in this study is that we have set up registries, and you can obviously do that in chronic diseases such as atrial fibrillation, where we have used registry to identify patients that can potentially enter our study, and therefore making sure that we locate center the right locations, or that we approach patients with the right patient characteristics in order to be included in the trial, and we do identify those patients in advance. The other thing that we are doing with this study as well is that we automating the processes of this study as much as we can in order to what I pointed out earlier, to improve the quality as well as the speed that it takes us to conduct these documents. I would like to finish my presentation, if you go to the next slide, please, by briefly touching upon what will happen in respect to data and what data we will look at and what data we will have available. We are right now in a world where we're very much focused, specifically, of course, for pivotal studies, on trial evidence. Trial evidence is a standard that is established around the world and is accepted as data that will help us to make clinical decisions, such as the approval of a drug in order to show efficacy and safety of a drug. Real-world evidence is being used already now, but real-world evidence is mostly used right now in order to look at outcome of different therapeutic approaches in the real world, mostly after, for example, drugs have been approved. What will happen is that the importance of real-world evidence is going to increase. Real-world evidence will be increasingly important to generate data, and we will increasingly rely on real-world evidence to make, for example, regulatory decisions around efficacy and safety as well. We will then find ourselves in the long run in a situation where we'll end another quality of evidence, and that is predictive evidence. So the world that we're looking at is going to shift from a more traditional approach of using mostly trial evidence to make decisions into a world where we'll use different kinds of evidence in order to make decisions. And of course, we can only use this kind of evidence if we are sure that we are collecting, processing, analyzing, and interpreting all this different kind of evidence appropriately. And we'd have to make sure that we have the systems in place in order to do that. And with this, I thank you for your attention, and I'll invite any comments you might have.
Thank you. Thank you so much, Mr. Christoph Koenen, for sharing that wonderful presentation and insights with us. Thank you once again.